It's called Seurat. Sorry. Yeah, sure. Okay, so just a little bit of the overview. So I'm gonna start by talking about the motivation of spatial transcriptomics data. And then we're gonna dive into actually the computational side for the skill seminar today. Specifically, I'm gonna talk about what is a Seurat object, how can we extract different things, different kind of information we're interested in. And then I'm gonna showcase like how to analyze the single cell RNA sequencing data or spatial transcriptomics data in Seurat. And lastly, if we have time, I'll also briefly mention some of the tips to convert data formats between R and Python. So, okay, so first of all, like here is a very nice analogy for spatial transcriptomics. So on the top, as you can see, this is like the 3D actual biological structure of the original organ. And then starting from the Human Genome Project in around 2000, all the way until like sometime before 2010, like people have been mostly using bulk RNA sequencing. So in that sense, we don't have the single cell RNA, single cell resolution. And then about like starting from 2013, there has been this single cell RNA sequencing technology so that we can also capture the sequencing data for each single cell to reveal the cellular heterogeneity in terms of different cell types or different cell states. But the problem is we still lost the spatial structure, which is what spatial transcriptomics is trying to do. And this is really a very hot and very new field that just evolved, like I would say 2 or 3, maybe at most 5, 6 years ago. Okay, so just to motivate a bit about why it's not sufficient to only have single cell RNA sequencing data. Okay, so here there are 3 different figures. Each figure corresponds to one tissue. And as you can see, each dot basically corresponds to one cell. And then different colors correspond to different cell types. So as you can see that the spatial distribution of cells in each cell type is not homogeneously distributed across the entire 2D space. Rather, there's some very complex spatial structure embedded there that are telling us something about the organizational structures. And this is what we want to obtain or infer from spatial transcriptomics data. This is just a zooming for the tumor tissue. You can see different cell types that are co-localized in different spatial regions. And so with all that, so there has been a lot of potential and opportunities in both the wet lab and the computational side. And so specifically for the wet lab side, this is a non-exhaustive overview, but mostly there has been 2 very popular branches of the wet lab sequencing technologies. The first is the top, basically it's using fluorescence in situ hybridization or sequencing approach. So a lot of different FISH methods have been proposed, even probably into this week. Yeah. And another method is based on the next generation sequencing method. Basically for that, what people do is as you can see, there's the spatial wall array, and then inside of each spot, there can be multiple single cells. So this is like the next generation based method, and a lot of methods also are proposed here. I think, for instance, SLICE version 2 was just published at the end of last year, and also for Transvisium, they're gonna have a Visium HD to be published next year. So a lot of exciting opportunities here. Okay, but no matter what kind of technologies we're using, at the end of the day, okay, there's a chart. Okay, awesome. Okay, so no matter what kind of technology we're using, at the end of the day, we're just gonna get the gene expression count matrix on the left that you can see. So this is basically a gene by location matrix. In terms of the location, it can be just the spots. And so besides this gene expression count matrix, we also have the two-dimensional, so X and Y coordinates for each of the spatial location that are aligned with our gene expression matrix. And then so basically these are like the two modalities of data that we have, and then we can do downstream analysis from that. And then hopefully you have already have access to the tutorial that I provided. Basically here, there is also a spreadsheet that I attached with different spatial data sets and some of the computational packages specifically designed for spatial transcriptomics. So yeah, some packages are in R, some in Python. Okay, so that's about spatial transcriptomics. Any questions so far?
Where was the data set or the tutorial?
Oh.
The tutorial's right there, sorry.
Okay.
Didn't announce that enough probably, but it's a shortened version.
Yeah, great question, thanks. Yeah, so hopefully if you can open the second link that Matt helped you right there, you will open this page that's on the screen, and then specifically like under materials, there's a HTML and also a R Markdown file. So if you wanna play with it by yourself, you can download the R Markdown, but for now I'm gonna just use the HTML for easier illustration. Okay, so yeah. Hopefully everyone has already opened their tutorials. Okay, so now I think I'll have to cut it in this way. Okay. So so I'm gonna start by talking about how to load packages and specifically the data sets. What is like sort of the data format for spatial transcriptomics data, and then we're gonna go through the analysis pipeline. Okay, so I'm gonna stick with dplyr instead of base R throughout the tutorial, but you can also feel free to try out base R. And then when we load the spatial transcriptomics data, you can directly download it from the webpage of Text Genomics. So here, just to make things easier for now, I have already downloaded the data set and uploaded them to Google Drive. So we're just gonna run through this code chunk so that we can directly download the data from Text, from the Google Drive, sorry. Yeah, so specifically, the most important thing I want to mention is that there are two data files that we need to download. The first is a feature by cell or feature by slot matrix. And I downloaded it as HDF5 file. And another is a spatial imaging data folder that is necessary for us to obtain both the spatial coordinates and also the actual imaging of the data. So we have to have both data files in order to do downstream analysis. And specifically, Seurat has this very nice built-in function called load 10x spatial. So with that two data files I mentioned, you can directly specify them and then download the data as a Seurat object. Yeah. Okay, cool.
Hey,
yes.
Another question. Could you describe what that slice parameter is doing? Like, what are the different, how is this different than the normal load 10x?
I see. Yeah, that's a great question. So, okay, so for the different arguments, the data.dir just specifies the directory in which we downloaded the two data files I just mentioned. And then file name should specify the name of the gene expression matrix. And then actually for the assay and the slice, these two arguments, they are just meant to specify the name inside of the name of the assay and the image inside of our Seurat object. So you can really name them as anything that you want. It's just, yeah, to make things more organized, I would say. Yeah, and then the last two arguments, they're sort of by default. Whether, so because tags provides like a filtered version and a raw version of the gene expression matrix. Yeah, just to clarify that. Yeah, great question, thanks. Okay, yeah, feel free to stop me whenever you have any question running the code. Okay, so now our Seurat object is the bring data, and then we can explore the Seurat object. So, okay, you can go through the details. So here, I think I'm just gonna highlight some of the things. So for the gene expression matrix, that's bring data and then add assay. And then inside of assay, there's, do I have it here? Yeah, so, okay, yeah, so for the gene expression data, that's inside of this spatial slot in the, among the assays. So if you wanna see, okay, how many assays, what are the different assays I have, you can just use names, and then it will give you all the named objects inside of this Seurat object. Specifically, yeah, this is how we can call our gene expression data using bring data spatial. So that's for the gene expression data. And then next thing that you might be interested in extracting is the sample level metadata. And that is a data frame. Specifically, we can extract it either using by using this double extract operator, or we can also use add met.data, which will give us like different sample level metadata. And one advantage for Seurat is when you load the data into Seurat, it will automatically calculate the total number of counts of detected molecules and the total and the number of features in each sample. So, and they will be very important when we do the downstream quality control. Right, any question here? Okay, cool. So that's the sample level metadata. And another thing is the feature level metadata. So this is assay specific. So by assay, we can imagine, okay, maybe you have the RNA data, you have the, which is gene by cell, maybe you have the spatial data, which is gene by spot. We can also have the ADT data, which is like basically the protein level data obtained from CySeq. So, or ATAC-seq data, basically any kind of different assays can be stored in different slots inside the assay part. And then this feature level metadata, it's assay specific. And if we wanna extract the feature level metadata, then basically we have to specify the assay of interest. So for instance, here, yeah, I just look into the met.features inside my spatial assay. This is how I get the feature level metadata. Okay, so that's all basically all about like the Seurat object. Is everything clear? Okay, great. Cool. So now I'm gonna dive into the analysis pipeline in Seurat. So the first two major step is pre-processing, and then do some sort of like the traditional downstream tasks. These two major steps, they can be generalized to either single cell RNA sequencing data or spatial transcriptomics data. So for pre-processing, the first thing we need to do is to do quality control. And some of the common QC metrics include like the number of features, number of counts, and also the percentage of reads that are mapped to the mitochondrial genome. And so we can, if the percentage of the reads that are mapped to mitochondrials are not provided, then we can use this percentage feature set to calculate that to calculate that sample level metadata. And then we can visualize these metadata using violin plot, using this function, which will tell us like how the data looks like. And then when we actually do the quality control, it's recommended to jointly consider all the 3 or even more metrics rather than looking at them one by one. So, okay, if we look at it using this feature scatter function, then here we can see the pairwise relationship among the 3 QC metrics. And then we can do the filtering or do the quality control using this subset function in Seurat. Yes.
Could you give us a sense for what exactly you're looking for in these plots to determine which cells are low quality, which cells are high quality?
That's a great question. Yeah, thanks. Sure. Okay, so then if we look back at here, so for instance, for the percentage of the mitochondrials, it's normally better when this percentage is small, because if it's too large, that indicates probably the cell is dying. And then in terms of the number of counts, we don't want it to be too small or too large. If it's too large, that might indicate the data or the sample that is collected is most likely to be not a single cell, but rather a doublet or triplet. So we don't want that. And also similarly for the number of features. So we don't want them to be either too large or too small. So for instance, So for instance, here in terms of the pairwise, when we look at the number of feature versus number of counts, probably I don't want these samples on the very right of the tail, and also the ones on the very left. Cool, great question. Yeah.
Where do you set the cutoff for something like the percent MT? That's kind of a general idea.
The percentage, sorry for what?
MT, the mitochondrial.
Oh, yeah, that's a great question. So mostly, yeah, what I always do is to look at the empirical distribution for this. So for instance, here, yeah, I feel like maybe a 30% threshold is sort of, yeah, acceptable. Or if you want to be more strict, then you can set it to be 20, but then you also sacrifice more of your samples. Yeah, the sample size will be smaller. Yeah.
So is it fair to just, you're basically looking for when it thins out and where you just empirically are seeing more outliers?
Yes.
Okay.
Yeah.
So
Yeah.
just, it's like a by eye or the outliers.
Right. Yeah.
Okay.
That's normally what I do. Yeah. Cool. Okay. And all right. So we can use the subset function to do the QC, and then specifically inside of this subset argument, we can specify like all the criterions for like, okay, which set of samples I want to keep. Okay, so that's about QC, and then after QC, another another pre-processing step is the normalization. So, okay, so here I first plot QC matrix on the 2D space using the spatial feature plot function. QC matrix, they also have their, sorry, yeah, just, they also have their spatial heterogeneity. So previously, if you're familiar with traditional, is want to normalize so that all the samples, they basically have comparable library size. But here, as you can see, this does not apply for spatial transcriptomics data because for their QC metrics, we should not expect them to be sort of uniformly distributed across all the different spots. Rather, like such heterogeneity does tell us some true biological signal rather than pure technical noise. So, so that's why, like Seurat team, they propose this SC transform rather than the traditional log transformation. Okay. And so in order to do the SC transformation, we can use this SC transform function. And so basically, if we do that, that will create a new assay called SCT. And all of the normalized data will be saved in that assay. But one thing I found can be tricky is inside of this assay, there are actually three different slots storing like the gene expression matrix. So the first is called counts, which basically saves. Yeah, so I think I have them here. So for the counts slot, that saves the corrected UMI counts. So that's the count data. And then the data slot, that saves the log normalized version of the corrected UMI counts. And then lastly, there's also a scale.data, which does some extra things. The first is that, okay, so what it actually stores is the Pearson residuals. And it's also used as the PCA input. And also, so I think by default, it is already centered, and we can also specify whether we want to scale it or not. And the last thing is that if we look at the dimension, this is, by the way, in R, so it's the number of features by the number of samples. So we can see actually the number of features in scale.data is different from the other two slots. And that's because in scale.data, they only return the top variable genes. And by default, that number is 3,000. So this is also one optional step in the pre-processing. Okay, any questions so far? Yes.
Can you repeat what you just said for the scale data with the number of features?
Sure, yeah. Okay, so the question is, why is like the dimension for the scale.data slot is different from the dimension in the other two slots? And the answer is that, so when Seurat is trying to calculate scale.data, automatically they set this return only variable genes to be true. So for scale.data, they will only return the top variable genes. And that number of variable genes is 3,000 by default. Of course, we can also set this Boolean argument to be false, or we can increase or decrease the number of top variable genes we're interested in. So yeah, that's like a small caveat. All right, nice. So after pre-processing, now let's look at some of the downstream tasks. So in 3.2, all the analysis steps can also be applied to single-cell RNA sequencing data, so without spatial coordinates. All right, so the first thing, by the way, yeah, as I mentioned, so we're just gonna stick with our normalized version of data in the downstream analysis input. So, okay, the first thing that we do is we run PCA using this run PCA function, and then you can compute the k nearest neighbors, and then do Leiden to detect different communities using this find clusters function, and then run UMAP. So now inside of our Seurat object, this is the most recent Seurat object. It will contain like all these analysis results, because one Seurat object is sort of like a container of both our data and all the analysis results inside of the Seurat pipeline. So yeah. So okay, now our UMAP result is also saved inside of this Seurat object, and I can look at and so I can look at this two-dimensional embedding from UMAP, and also I can annotate the clusters that we just identified. We can annotate these clusters back onto the 2D space. So the first thing, like the left plot, can be obtained using the dime plot function, and then the right-hand side, these plot with the spatial coordinates can be generated using the spatial dime plot function. And the right-hand side, this figure, the figure on the right is spatial transcriptomics specific. Okay, and so yeah, just want to mention where were these analysis results saved. Basically, inside of the Seurat object. So besides the assays and some of the other slots that we just saw, there is also another one called reductions. And then if we look at reductions, okay, right now it has two different slots. The first is called PCA, the other is UMAP, and they contain all the relevant analysis results or information there. Okay, and okay, since we have already annotated the clusters, if you want to take a look at those annotated clusters for each sample, then you can specify this active.ident. This will tell us like all the labels that we just generated. Okay, so and then another downstream task that we can do is to do differential expression analysis. So here I want to mention, okay, this is independent from the spatial coordinates. So we're just using the gene expression data to do differential expression analysis between like different clusters that we just identified. And we can do such differential expression analysis using this find markers function. And so, yeah, sorry, I think there's a typo inside of the comment. So for this line, I'm trying to find all the markers of cluster 10. So what I'm doing here is by specifying ident.1 is 10, I'm trying to find all the markers in cluster 10 compared to all the rest of the samples. And if you, for instance, if you want to do the DE, the pairwise DE, so look at the marker between cluster 10 with cluster 11, for instance, then you can specify ident.2 is equal to 11. Okay, and then, yeah, we can do some sort of plotting using the functions that we already saw previously. Okay, and now next, yeah, I want to sort of using the next 15 to 20 minutes to talk about some of the analysis that are specifically for spatial transcriptomics data. And here specifically, yeah, I want to talk about how to identify the spatially variable genes. If you recall, like just now while we were talking about the DE analysis, we did not use any of the spatial coordinates. Okay, so but here we're gonna actually also incorporate the spatial coordinates as our input to identify the genes or the features which are spatially variable. So I'm gonna mainly talk about the methods or the approaches provided by the Seurat package, but there are also a lot of other packages that do similar thing that I'm gonna mention after this. Okay, so for Seurat, they provide two different options to do this, or two different metrics. The first is called Moran's I, the other is called marker variogram. Okay, and in order to calculate these metrics, we need both the gene expression data and also the spatial coordinates. So, okay, let's take a quick look at what Moran's I basically it's about. Yeah, you don't have to read all of these words. It's too much here. But basically, okay, there's a chat. Sorry, yeah, never mind. Yes. Okay, yeah, thank you. Okay, yeah, so intuitively, Moran's I is just a spatial version of the Pearson correlation coefficient. The way to understand, the way to think about it is basically is trying to measure the correlation of the gene expression values between the local observed values with its neighboring, with the gene expression values of its neighboring samples or cell spots. Okay, so where's my mouse? Okay, sorry, yeah, just trying to find my mouse here. Okay, yeah, so yeah, I just also provided the math equations for Moran's I and also the Pearson correlation so that you can see that they're actually quite similar. And yeah, just to explain the notations, N stands for the total number of spatial spots, and then Xi and Xj, they're the gene expression value in location I, J for one specific gene. So Moran's I is like a gene-specific metric. And one very interesting in this W basically encodes our prior assumption about the spatial information, which I'm gonna briefly introduce later. But here, yeah, just to give an intuitive sense of what Moran's I is, so it will be positive. So it's somewhere between -1 to 1. Okay, so it will be positive if the similar values they cluster together. So if we can see some very clean, some very strong spatial relationship, then that's positive Moran's I. If there's no spatial autocorrelation, if like there's no spatial structure, then the Moran's I will be 0. And if the similar values are separated from each other, this will be called as, then our Moran's I will be negative. Okay, so that's like the intuition about what Moran's I is. And then for the weight matrix, that's basically a way to try to encode our prior assumption about the spatial neighboring relationship. So there are two ways that we can do this. The first is distance-based. Basically, the W sub ij can be an inverse of the distance between slot I and j. So that's the distance-based, or it can be contiguity-based. So if the two spots, they share one boundary, then we can encode that as 1. Otherwise, it can be 0. And also, so that's only the first order contiguity. We can also make it to be higher order contiguity, or yeah, for instance, to have this sort of decaying function. And yeah, and then specify that back into our Moran's I calculation. Okay, and then so that's like a brief introduction about this metric, and then specifically we can calculate Moran's I using this find spatially variable features in Seurat. And so, okay, we need to provide like specify the assay, Seurat object, and also, so here for the features, this is like the features or the genes to be considered in the analysis. So here, just because of the computation cost constraint, I only look at the top 10 genes, top 10 variable genes, but you can actually look into all the thousands of genes. And then specifically for the selection dot method, we can specify that as Moran's I, which is how we get to specify this. So where's the actual output? So the output of Moran's I is saved inside of the assays object. So in SAT slot, we can look into the met.features. And because I only look at the 10 genes, so the rest of the genes will be NA, so I excluded them with dplyr. And then, so this is the an example of this is the result that we actually got. So for each row corresponds to one gene, and we have 4 different columns. The first is the observed statistic, the Moran's I. The second is the p-value for the Moran's I under normal assumption. And then the third is like a Boolean argument about whether this gene is indeed spatially variable or not, and that's based on the p-value. And lastly, they also ranked the spatially variable genes among the gene list that we gave to Seurat, and such rank is ordered by the observed test statistic, so which is Moran's I. Okay, and then yeah, this is how, and then in the next chunk, this is how we actually visualize the analysis result using the so-called spatial feature plot that we saw earlier. And yeah, I just also want to mention that this plot annotation function is quite nice when we want to combine different ggplot. So, and this plot annotation is from Patchwork, like another package. Okay, so basically here, these are the 3 genes out of the 10 genes that I considered. So these 3 genes, they are they have the largest Moran's I statistic. So as you can see, indeed, they have some very strong spatial structure. And I also look at the very last 3 tails, just to see whether there's any difference. And indeed, as we can see here, even though, I mean, so actually for all the 10 genes that we looked into, all of them are spatially variable. But you can see that even though they're also spatially variable, the bottom 3, their spatial structure is not as strong as the top 3. It's not as obvious as the top 3. Okay, so that's about Moran's I. And then for another metric is called marker variogram. And this is, so variogram basically it's a function with respect to the pairwise distance between 2 slots. So if we specify the pairwise distance of 2 slots as R, then the variogram of R is the average of it's an average of the squared difference of the gene expression in 2 slots. And also, I mean, variogram is also gene specific. So for instance, xi and xj, they stand for the gene expression value for this specific gene in slot I and j. Okay, and then we look at its square difference. And because within the whole 2-dimensional space, there can be multiple pairs of slots of the same distance r. So then we take the average. And that's like the variogram that we're interested in. And okay, so then we can model the marker variogram as this sort of function. So the y-axis is the variogram, and the x-axis is the distance r. Okay, and then we can use statistical modeling to see whether such association is indeed statistically significant or not. And the only difference is, yeah, in selection.map method, we can use marker variogram. Okay, and then, yeah, so I'm doing something very similar here, very similar to Moran's I. And you can see, okay, here are the 3 genes, the top 3, the 3 genes with the top spatial variability identified by marker variogram. And then here, down here, these are the 3 bottom ones. So indeed, yeah, again, we can see that the ones on the top, they're stronger, and the bottom ones are a lot of weaker than the previous ones. Okay, so that's about the 2 methods. But one thing I also want to mention is that we also need to keep in mind that they're not actually trying to identify the true spatially variable genes. I mean, it also depends on our definition for spatially variable genes, but these genes, their spatial structure is actually probably very likely to be explained by the spatial location of the cell types. So for instance, for the MBP gene, maybe such spatial heterogeneity can be largely explained by the cell type heterogeneity. So a more ideal way to identify spatially variable gene is to first constrain our gene expression matrix and only look at the and only look at the samples of the same cell type. And then we can try to identify the genes with purely spatial heterogeneity. So this is also like a research field that's under investigation now, which is also very exciting. Okay, so that's about how Seurat can identify spatially variable genes. Are there any questions so far? Okay, cool. Yes. So then I want to quickly mention that there are also some other packages in R that can identify spatially variable genes. All of them, have very good documentation. So hopefully it'll be easy for you to try some of them. Yeah. So for spatial D and transient, they were published in 2018, Spark in 19 or 18, and then Moring is like from Jing Fan's lab. Yeah, I think it was published last year. Yeah. Okay, and then lastly, I also want to point out some of the toolkits to convert data formats between R and Python, because I know some of you guys might be a heavy Python user. So the two packages that are mostly helpful is Seurat disk and SE-easy, and another like probably more sophisticated one is the reticulate package. But with this package, we can also run Python script from R. So that's another plus for this. Okay, I guess that's okay. Any questions so far? Yeah, cool. I guess that's all I have for now. And I just also want to mention that, so this tutorial is largely based on the computational assignments that I created with Yun in graduate level class that we're offering this semester. And a lot of things that I mentioned just now were based on the Seurat tutorials. And also, special thanks to Sawan for feedback. Okay, cool.
So first off, thank you so much for that really good tutorial. This especially R Markdown file, I know I'm going to be very glad to have if I ever do test analyses. We have about 5 minutes left until 1, so perfect timing. Just a couple of items before we jump into questions. There's a feedback form right there. We'd really appreciate if you fill out that feedback form. Lets us know what you want to hear about. It helps us get